Okay, I'd like to begin by thanking the organizers, especially Sashi and the Academy as well as the India-US Forum for inviting me to this meeting. As the first two speakers already pointed out, many aspects of what I'm going to be talking about, my work has been made easy because hopefully by now you know what a network is. But as Adam pointed out, I'll be taking a much more larger scale view of what Trey has so far been talking about. As you probably inferred from my affiliation, I'm not a biologist. I'm trained as a physicist, and a physicist has often been compared to some guy who's looking at the world, but is gradually defocusing so that details all get washed out, and what he's seeing are the broad patterns. So essentially, what I'll be focusing on is looking at the unifying themes, the universal patterns that appears in networks that occur at very different scales. So here, right over here, you have the smallest scale, the intracellular signaling network. And over here, you have the largest scale possible, at least on Earth. This is the food web, a particular ecosystem. So here you're talking about molecules in the cell. Here you're talking about species which populate a particular patch of the world. And the timescales are also very different. I mean, here we are talking about seconds or even smaller time scales. Here we are talking about generations. So what we are interested in is find out what kind of patterns bind these very different systems together and also systems intermediate. So in case you still haven't got what a network is, I just thought I'd hammer the point home once more. So a network essentially consists of nodes. So these nodes may be molecules, or they may be cells, or they may be species or individuals, depending upon which scale you are looking at. And they are linked by interactions. So this could be, like in Trey's system, these are protein-protein interactions, but this could also be cellular communication links, or this could be individuals who are coming into physical contact with each other, or species which have trophic relations. And this combination of nodes and links is what you call a system. And what I mean by network biology, as already has been pointed out by Adam in his introductory talk, is network biology is looking at how this interaction between components give rise to features at the system level which you don't encounter at the level of the individual components themselves. Let me give an example. So if you look at the brain, which is made up of billions of neurons, now these neurons don't necessarily show the property that you usually associate with the brain, namely conscious thinking. However, you put a billion neurons together, connect them densely, and out comes a system which is actually giving this talk. So no surprises over here, because you all know that interactions can add layers of complexity on what is there already in this component itself. And network biology in particular wants to study how, as a result of these interactions, you have this complexity, which somehow pops out through emergence. The particular kind of complexity emergence that I'm interested in is, in particular, how does robustness appear in systems, in particular biological systems, as a result of this complex set of interactions. Now, we all know that living systems are stable, and yet they can adapt to changing circumstances. So in some sense, they are complex, yet they're robust. So one of the biggest challenges to us is to find out how can you build systems which have this property of complexity, yet being robust. And from this point of view, rather a blanket point of view, I look at systems biology as essentially network biology. And such networks actually appear at all scales in the biological world. At the smallest scale, you have the intracellular network. I won't be speaking much about this since both Adam and Trey have spoken about this in detail. But we have been doing some work with biologists at Delhi and at Pune on the B-cell response signaling network. So B cells are involved in the immune response system, and what this system is involved in is giving out the correct response to certain kind of stimuli. So a breakdown of communication in this particular system essentially leads to disease. So obviously it's very important from also clinical and medical point of view to find out what can lead to breakdown of communication among the constituents of the system. So of course, this system as a whole is very hard to study. So normally people kind of focus on a subset of this system. So here is a subset that, for example, our experimental colleagues are focusing on. So most of these molecules are kinase proteins which activate other kinase proteins. And this here is P38, which actually Gopal, I think, mentioned in his talk. So which is involved in the cell committing harakiri. Now, what the experimenters do is, for example, they would block the activation of this particular molecule and see how this affects other molecules in the system. Now, from this picture here, you would think, well, I mean, if you block the activation of P38, that shouldn't affect what is going on over here, because P38 is at the end of this chain, which is somehow standing completely apart from the rest of the system. But to initial surprise of many, it turned out that when you blocked activation of P38, it tended to impinge on the activation of several other protein molecules here, which kind of led to an understanding that this wasn't the correct picture of how the individual components interact at all. So what is happening is that there are lots of missing links over here. It's actually a quite densely connected network, and one of the first tasks that we had was finding out these missing links. And once we find the missing links, then we want to answer the question of how does this network allow the cell to be very differentially responsive to different kind of stimuli. So depending upon what kind of infection occurs, it will respond very differently. And yet, it's robust enough that even standing in this extremely noisy environment of the cell, it can respond correctly most of the time. Then, if you go up one step above, we get these intercellular networks. So, for example, neural networks is one such example. And in the case of this worm, C. elegans, you actually have one system where the entire neural network has been mapped out. So this is a kind of schematic of the neural network where the nodes stand for the neurons, and they're all linked to each other. Now it looks fairly, kind of a random ball of multicolored wool. But one of the things that we have been doing is looking at the hierarchical organization of this network. And when you look at it in detail, it turns out that it's far from random. There are organizing principles that are clearly at work, very systematic organizing principles which make this very far from a random network. You can, of course, also look at a slightly higher up level where you're looking at the different tissues, organs in an individual, how they interact to produce a functioning individual. But in the level above that, you can have a network of individuals who are in contact with each other, and then you want to study how infections can propagate in this network. So, for example, suppose you have a individual contact network like this, and you make this individual infected with a particular disease, and then you want to find out how this disease propagates. So, this kind of studies are extremely relevant if you want to have an intelligent way of vaccinating a population against, say, maybe SARS. So, it turns out that instead of just homogeneously trying to give the vaccine to as many people as you can, it's probably much more effective if you can first inoculate the hubs, or the people with the largest number of contacts. You somehow find them out, inoculate them, and that way you'll be able to combat this spread of the infection much faster than you'd be able to do otherwise. And at the highest level, in spatial as well as temporal level, you have the ecological networks. So, for example, like food webs. So this is an example of a particular aquatic food web occurring in the ocean shelf. So here is the kind of dividing line between the fish, which the different species of fish which are appearing here, and the birds, and species which feed on each other are essentially linked by arrows. Now, the question that we are interested in this scale is, for example, why do ecosystems collapse? So it often turns out that essentially extremely small fluctuations can trigger mass extinctions, which may cause a whole ecosystem to collapse. And this, in particular, assumes a lot of importance in view of human interactions in ecosystems, when many ecosystems have become extremely fragile due to the way humans have affected it. And one of the particular questions that people are extremely interested in, both from a practical as well as a theoretical point of view, is, for example, is higher diversity in the ecosystem good or bad in terms of stability of the network? Okay, so the research program that we are engaged in is essentially looking at how does network structure affect network dynamics, and also in turn how does dynamics constrain the kind of network structure that you see in nature, and how do both get affected by the functional criterion that this network has to satisfy. So as the first step, we kind of look at how does network structure affect the dynamics, and as an example, I wanted to show a particular kind of, a particular class of network structure and how this leads to a very novel kind of dynamics. So the example I had in mind are the so-called small world networks, which is kind of an interpolation between two very familiar class of networks. At one end, you have this regular network, essentially whose components are connected in a very localized fashion, nodes being connected only to their nearest neighbors and so on. At the other end, you have random networks where essentially you pick up any two nodes at random and link them with certain probability. However, in between them, you can have a small world network which has certain properties of a regular network. So most of these connections actually occur between nearest neighbors. However, with a very certain small probability P, you can have long-range links. Now, the people who first proposed this network model actually claim that there are many systems, both technological as well as natural, in particular the C. elegans neural network, which shows this kind of small world behavior. But the question was, okay, it's a good model, but how relevant is it to biology? So it turns out, there have been recent reports that there are several brain functional networks which were obtained through magnetic resonance imaging and so on, have claimed that such networks also seem to show this small world property. So we kind of try to figure a very simple system where small world effects can have interesting neurobiological consequences. And the particular model system we had in mind was a system where you have lots of glial cells, the so-called support cells in the nervous system, and neurons. So the glial cells, which are connected in a very locally fashion, essentially form this local matrix. So whereas neurons are actually acting as the long-range connections between these glial cells. And all the cells actually can communicate with each other through the calcium waves. So what you have is essentially a regular array of glial cells which are forming this very locally coupled group. And on top of that, you have neurons which are acting as long-range connections. And so together you have a small world network of excitable cells. Now, when we model dynamics on this kind of a system, we found very interesting behavior. In particular, we found that as we increase the fraction of long-range connections, that is, as we are changing the ratio of neurons to glia, we get a transition from very different kind of patterns. So if the number of the fraction of neurons is less than some critical fraction, then you have get spatial patterns which are very much like spiral waves, which Adam showed in his talk. However, the temporal behavior is extremely irregular. If you increase the fraction beyond this critical value, but it's still less than some upper cutoff, you get temporal patterns which are characterized by extremely periodic bursts, but which is spatially rather homogeneous. And finally, if you go beyond this upper cutoff, then you get failure of self-sustained activity altogether. Yeah.
Clarification
Yeah.
on the slide for this one.
Okay.
The one before. So, did you say the glia of the excitable cells, or what? I didn't
No, the glia actually communicate with each other through calcium waves.
Okay.
Okay.
And so the small world network is just the neurons communicating with glia, or
No.
the neurons among themselves? I mean,
No, the small world network is where the cells communicate with calcium waves. Okay. You probably got confused by this excitable word,
yeah,
right?
network of excitable cells.
Yeah, so excitable is not having this classic action potential. By excitable, I don't mean cells communicating action potential.
What do you mean by fraction of cells?
Okay, so essentially I mean what's the ratio of the neuron to glia. So here, in general, glias outnumber, in fact, as in reality, glias outnumber the neurons. So as you are increasing the fraction of neurons, you're keeping the glial cells fixed. How does the dynamics change? So to look at in detail what is going on, if you are below this lower critical value, then you get this spiral waves. whereas temporally you are rather irregular. So if you do the frequency, if you look at the frequency spectrum, this looks more like white noise. On the other hand, if you are between these two critical values, between the lower critical value and the upper cutoff, you get extremely periodic time response. So, the kind of activity goes up and down with a extremely precise frequency. However, spatially they look rather homogeneous most of the time. And then if you increase the fraction even beyond that, you just have transient activity where the activity goes up and then eventually dies out. So the summary of the first part of this work, relating structure to dynamics, is that you can have self-sustaining pattern formation in such small world kind of network, which exhibits patterns of two kinds, spatial as well as temporal, but depending upon what is your network structure actually like. And of course, this has relevance for many kinds of systems, but in particular, maybe for how non-trivial network topology affects brain function. In particular, we were excited by the connections they seem to have with the pictures that we see of epileptic seizures and bursts. Okay, on to the second step of this program. The more interesting part of this program is where we are looking at how functional criterion, in this particular case, the need to have stable network dynamics, how does that constrain the network structure that we can have? So this actually relates to a decades-old question about, are complex networks unstable? So for example, as you increase the complexity of a network in terms of how many nodes it has, what is the density of its connection, and the strength of interaction between the nodes, does the network become more or less stable? It first historically arose in ecology, but of course it has ramifications in all sorts of disciplines. So the big puzzle was that theoretical results imply that as you increase each of these quantities, it actually decreases stability. But if you look at the empirical observations, they seem to show the opposite. So there have been lots of criticism of this original theoretical results on the grounds that these were obtained assuming, first of all, the networks are random and that they were at equilibrium. So, very trivial kind of dynamics. So we took a fresh look at this whole debate by, introducing lots of complications in the origin formulation. So first of all, we looked at what happens if you introduce structures. So instead of random networks, if you have, like say, modularity, for example, how does it change this, or does it change at all, this proposition that complexity implies instability? We also introduced dynamics in this whole formalism and try to see whether it changes this, but both of these efforts showed that this actually turned out to be an extremely robust result. Finally, when we introduced evolution into this picture, we found that actually this complexity to instability result and the complexity to instability, which was claimed by empiricists, can be resolved in a very nice sort of way. So the first part, the thing that is how the structure affects stability. So for example, you can ask how do small world networks affect this complexity to instability result. It turns out that there's very little dependence of structure on this complexity implies instability result. All it does is that the stability to instability transition becomes sharper as you make the network more random. How about modular networks? Now, to define modular networks, essentially you say that a network which can be divided into subsets such that there are more connections or dense connections within each part compared to the others. So these extremely densely connected subparts you call modules. So this is an example of a food web, but you also see examples of modular networks in all sorts of areas. So for example, even in functional interaction among brain areas. So we were kind of looking at what happens in modular networks, but here also we found that in fact, interesting, what we found was that as you divide the network into more modules, they actually become more unstable. However, you do see modules in all sorts of networks in this particular case, the C. elegans synaptic connectivity matrix. So, obviously it must be stable somehow. So the question was how? So the clue that actually got us finally to possibly one correct answer is that many of these modular networks also have multiple hubs. So hubs are essentially nodes which have lots of links. So in a technical sense, we say that is the node with large degree, where degree is actually the number of links that a particular node has. So the hypothesis that we started out with is that instead of just trying to satisfy one constraint, real networks are optimizing between multiple constraints. So in this particular case, it's trying to minimize the number of links because each of the links has some resource costs associated with it. It's trying to maximize efficiency. In this particular case, how many hops it requires to go from one node to any other given node. And then finally, it's trying to maximize stability. So if you were just trying to minimize the link cost and maximize the efficiency, you would have ended up with a star-like network like this. However, it's extremely unstable. And that's because instability is actually related to the degree of the hub. The more the number of links that this hub has, the more unstable it is. So if you want to satisfy all three constraints, the obvious answer is to go modular. So what you do is you take a star-shaped network and break it up into many subparts so that each of these individual hubs now have a much lower degree than the original network. And of course, you'll be satisfying all these three constraints that you had started out with. And so instead of just looking at the star-like networks, which are obviously not very realistic, we also tried out many different combinations which are much more real-looking in some sense. And it works in all of these cases. So but still, the bottom line is that even if you introduce the structures, the original result, complexity implies instability, does not change. So next we tried introducing dynamics into the nodes, and we tried out very different kinds of dynamics from fixed point to oscillatory to chaotic. And although we found interesting results, so for example, we found that the size of active nodes is extremely robust and the number of links is also characteristic, it still didn't change the original result that increased complexity implies network instability. So we were still dealing with a puzzle that how can you have complex networks in nature at all if they are so fragile? So the possible solution suggested itself to us when we were thinking of network evolution, in particular that all these networks, they don't just occur suddenly, but they gradually evolve over time, starting from maybe a few nodes and then gradually more and more nodes arrive, and that's how they kind of gradually grow over time. So a particular example we had in mind was this assembly of ecological communities. Now this is a picture from a book of Edmund Wilson, and so he compared this assembly of ecological communities with like building a jigsaw puzzle together. So how you build this jigsaw puzzle together pretty much depends upon which piece you find first and try to fit with what is already there. So for example, if you fit this piece to this puzzle, obviously you won't be able to fit this piece, and then this piece also won't be getting into this puzzle. So who comes up to who pretty much decides how this system will behave. So this actually can be theoretically shown by a very simple model where essentially we are building up a network subject to it satisfying the stability criterion at all times. So you start with one node, and then you add another node with a random number of links and also randomly distributed connection strengths, and then you check the stability of the network, and if it's unstable, you remove a node, and you analyze the stability again. And if it's stable, you add another node. So it's an extremely simple mechanism. However, the results are extremely surprising. So what you find is that the network initially does grow in size monotonically until it hits an instability regime, and then you get this series of growth and extinctions. And it settles down to a pattern of growth spurts and collapses around some average value. Now,
what is the measure of stability?
yeah, so in this particular, can I actually answer that during the question now, because I need to go into technical. So the big surprise here was that for these evolved networks, complexity did imply robustness when we measure robustness in terms of variability, that is how much the network size fluctuates around the average value. It was more resilient. So for example, after a fluctuation above the average, it quickly, much more quickly came back to the original size if it was much more complex. And finally, if you look at the extinction cascade, so what was the number of species that were lost during one whole extinction cascade, it was much more, it was much less probable of having a large magnitude collapse in a large network compared to a smaller network. So actually, you had increased resistance to these more complex networks. So the relevance for the second part of how dynamics constrains structure is that introducing dynamics and or structure into networks does not change the likelihood of a network to become unstable at increased complexity, although you can explain certain features like modularity. However, when you introduce network evolution, you can evolve networks which are at the same time complex yet robust. So this results, in some sense, tell you that the traditional picture of looking at networks at a single point in time, just taking a snapshot, may not be adequate for building an understanding of their robustness. And of course, there are implications from cell to ecosystems. So I'd like to end with thanks to my collaborators. So of course, this work has been done both with people who are working on the network aspects of it as well as in the biology aspects of it. So people over here are mainly physicists, and here Sir Chris is an ecologist at UCSC. Kanuri, Dheeraj, and Bhaskar are cell biologists who are working on the B-cell response network that I was talking about. Nived is a neurobiologist who is collaborating on the C. elegans work.